Okay, so welcome back to this next video in which we are discussing the major histocompatibility complex. Okay, so I'm just trying to explain the concept of a haplotype to you. Okay, so this concept that not all of the combinations are going to occur in nature, this is not the concept of a haplotype. The concept of a haplotype explains why only a few combinations are actually seen. Okay, so in order to understand this, we need to look at the process of meiosis, okay? So the process by which we create variation, basically. So, let's say, originally, that we start off with a human population where only these three combinations are present, okay? And we want to try and construct some new combinations, okay? So how would you construct new combinations? Well, the main way that you construct new combinations in other places in genetics would be in meiosis, basically, when you have crossing over of the DNA, okay? But this is not going to happen in this case, and this is because of this being a haplotype. Okay, so let me explain the process of meiosis, and then we can go further. So, you start off with a cell, okay? And this cell has 46 chromosomes within it. So I'll draw 3, but remember that there are 46 in reality. Okay, now what's going to happen is firstly the cell will copy all of these chromosomes, okay? So it will replicate every last one, and what will happen is that the exact copy will end up attached to the original one, so you'll get these sister chromatid structures like here. Okay, right. Now what we have to understand is that in this original set of 46 chromosomes, what there were actually 23 pairs, okay? So there were 23 pairs of homologous chromosomes. So, what's now going to happen is the homologous chromosomes are going to find each other, okay? So each of these three is going to find its homologous chromosome. So I'll put them here. Okay, so they're all going to find their homologous chromosomes. Okay, so this would go on for all 23 pairs, basically. Obviously, it would be more difficult in the sex chromosomes, because you're not going to get homologous recombination with X and Y, for instance. If you're a male, then you'll have an X and a Y chromosome, and homologous recombination does not occur across the sex chromosomes, the different sex chromosomes. Okay, right, but for the other 22 chromosomes, what's going to now happen is the two homologous chromosomes are going to line up, and they're sister chromatids now because they've got exact replicas of one another, and they're going to have crossing over. Okay, so let me draw a big picture of this. So, basically, we're going to need some colour to understand this. So, let's colour in this one, which is our first chromosome 1, which has these two sister chromatids. So there are really two identical copies of the same chromosome here. Okay, and then again, here's our other chromosome 1, okay, which we'll colour in green here. Now what's going to happen is bits of this chromosome 1 are going to exchange for bits here, and it's the equivalent bit. So for instance, you might take this bit, cut out the exact equivalent bit here, and swap them over. Okay? Right, so let me draw this now. So here are our two chromosome 1s again, and now that crossing over has been done, what you'll have is you'll have a little red strip here, and you'll have a little green strip here, and then the rest of this will be green, so let me put this on here. Okay, and then the rest of this will be red. Okay, so this process of swapping portions on one chromosome 1 for the portion on the homologous chromosome 1, this is what is known as homologous recombination. Okay, or swapping over at the chiasma, so homologous recombination. Okay, right. The portion where you swap over, that's known as the chiasma, basically. Okay, this process is also just called crossing over. Okay, so what you could imagine then is that if we looked at these two if we looked at the MHC class, if we looked at the major histocompatibility complex, so let's say these are now chromosome 6s, and we're swapping over bits from the two chromosome 6s, then you could imagine maybe that we could swap the HLA-DR maybe, okay? So I could take this HLA-DR out of one of them and swap it for the HLA-DR maybe on your other chromosome. So let's say on one chromosome 6, you have this MHC locus, okay, and then on the other chromosome 6, you have this MHC locus, okay, and you can imagine that what could happen is you could chop this bit out of one and put it onto this one over here and put this one from green into red, and then you'd have created a new MHC loci, because what you now end up with on the green chromosome is that you'd end up with 211,321, which is a totally new MHC locus. Now, this does not happen, okay? No. If you are going to have crossing over that involves the MHC locus, you basically move the entire locus. So you chop here, and the entire thing is cut out and swapped for this one. So you do not chop within the MHC locus. So you can't just exchange bits of the MHC locus. If you exchange the whole MHC locus. And that means that the two MHC loci you end up with are exactly the same as they originally were. So you don't end up with any new combinations. This concept that they are always crossed over all together, that is the concept of a haplotype. Okay, so they're inherited all together, and that's why you only have certain combinations. So your combination of genes will be called your haplotype, okay, but there are only certain haplotypes, and the reason there are only certain haplotypes is that you don't have this swapping over within this locus, basically. If you swap the loci, then you swap the entire loci. You cut here and here, here and here, and you swap them, which is why only certain haplotypes are allowed. So, the haplotype is not really this concept that they're only cut together. It's the concept of the combination. So you'd call the combination that you have your haplotype. So your combination of all of these HLA genes, that's your haplotype. The reason that there are only certain haplotypes, and why this is an important concept here at all, is that they cross over together, basically. You can't just cut out an HLA-DR gene and swap it for the HLA-DR gene on the other to create new haplotypes. That doesn't work. So only certain haplotypes are seen. You wouldn't see all 216 haplotypes. Now, in reality, there are many, many more of each of these. There are more haplotypes than 3, okay? But there aren't as many haplotypes as there could be, nowhere near. And this is the reason why, that the whole thing is inherited together. Okay, so just to finish our picture of meiosis then, what then happens is, once you've had homologous recombination, what happens is you're going to split up the homologous chromosomes, okay? So you line up all 23 pairs of the homologous chromosomes and you split the cell into 2, okay? So now each one of these cells has 23 sister chromatids, okay? So you don't break up the sister chromatids initially, you break up the homologous chromosomes. Okay, so they line up to do the homologous recombination, and then afterwards what you do is you split them apart. So 23 go in here, and 23 go in here. Okay, and there's also a process known as independent segregation here, because it's not as if all of the paternal chromosomes go into one and the maternal chromosomes go into the other. Some of the paternal ones will come in here, some maternal ones will come in here, some paternal ones will come in here, some maternal ones will come in here. So each cell then gets one chromosome 1 even though of course it's a sister chromatid, so you've got two identical copies of that, but basically they're identical, so effectively you only have one chromosome 1, because if you've got two identical ones, that effectively only counts as one, because you don't have two different alleles basically for each gene. Okay, so there's a process known as independent segregation, which basically means that it's random whether the chromosome 1 that this cell gets is the maternal chromosome or whether it's the paternal chromosome, okay? And if you might get the maternal chromosome 1, then the paternal chromosome 2, the maternal chromosome 3, etc. Okay, so independent segregation is the process of splitting up the homologous chromosomes, and then what you do is you break apart the sister chromatids, okay, in each one of these cells to then end up with cells which contain 23 chromosomes, and these are not sister chromatids, so they're not connected to an identical copy of themselves. Okay, so here we go. And these are all then haploid cells. Well, these are haploid as well, really. They may have an identical copy of themselves, but they don't have two copies of the gene, really. I don't know whether they would be referred to as haploid, but they don't have two copies of each gene. They have two identical copies of the gene. So I suppose they do have two copies of the gene. I don't know whether they'd be referred to as haploid or diploid. Okay, and then this one would split into two daughter cells as well, which would each be haploid again. So they'd have 23 chromosomes, and these would not have sister chromatids attached to one another. Okay, right. So that's the process of meiosis. So that's the concept of the haplotype of MHC. The haplotype is the specific combination of each of these 6 genes that you have. So it's the name for this combination, basically. You have a specific HLA-DP, a specific HLA-DQ, a specific HLA-DR, a specific HLA-B, a specific HLA-C, and a specific HLA-A. And there are only certain haplotypes that are actually seen in the population, and the reason you don't end up with every possible haplotype is because the whole thing is inherited together. So you don't get this switching of the different HLA genes within the MHC locus. Okay, so that's the concept of the haplotype, okay, and the fact that it's inherited altogether. Now, I just want to briefly mention the HLA DR MHC class 2 molecule that is involved in rheumatoid arthritis and gives you increased susceptibility to developing rheumatoid arthritis. So if you have the gene HLA DR4, okay, which is a specific allele of this HLA-DR gene, although of course we know it's not really a gene, it's a collection of genes, okay, then you will produce MHC class 2 molecules of the DR form that are specifically DR4, and basically if you produce this form of MHC class 2, you are 4 times more likely to develop rheumatoid arthritis than someone who does not have an HLA-DR4 allele. Okay, right, and that's going to be important when we come on to discuss the pathology of rheumatoid arthritis. Okay, right, one final thing that I want to discuss is how the peptides are actually bound to the peptide binding groove. Okay, so what does MHC class 2 recognize then? How does it decide which peptide should bind to it? Okay, right, so basically the MHC molecules, whether it's MHC class 1 or MHC class 2, they are not specific for antigen fragments. They are not like T-cell receptors. T-cell receptors are specifically complementary to antigen fragments. MHC molecules combine a huge variety of different antigen fragments. So we'll do an MHC class 1. To have a nice simple example. So let's say this is the peptide binding groove here, okay, and the peptide that lies in here. So how do you decide which peptides can bind here and which can't? Well, basically, in order to bind, each different MHC molecule will require a different bunch of what are known as anchor residues. Okay, so basically for each different MHC class 1 molecule, and remember there are 3 different types of MHC class 1, and for each of these different types of MHC class 1 there are absolutely loads of different alleles, so there are a huge number of different MHC class 1 molecules you can make. So let me illustrate this with an example. So we'll take the example of an HLA A3 MHC class 1 molecule. So this is an MHC class 1 molecule of the HLA-A form, and specifically it's HLA-A of the 3rd type. Okay, now there are certain anchor residues that are needed along this polypeptide. So let me split the polypeptide up. Let's say it's a 9 amino acid peptide fragment here. So we've done 6 now, 7, 8, 9. Okay, and basically you don't have to have a specific combination of amino acids, but you have to have certain conserved amino acids at specific places. So for instance, at position 2, there is a very specific amino acid that you have to have there to be able to bind to HLA-A3, and that is isoleucine, or I for short. Okay, then at position 5, so here's 3, here's 4, here's 5, you then need to have the amino acid phenylalanine, the single letter amino acid code for which is F. And then finally, at position 9, you then need to have the amino acid lysine. Okay, so position 9, which has the single letter amino acid code K. So let me just show you the structures of these amino acids. But before I do that, let me just discuss the concept again. So these 3 residues are the anchor residues for HLA-A3. For different HLA molecules, you'll have different anchor residues. So basically, I do not care what you put in position 1, 3, 4, 6, 7, 8. But as long as you have these conserved amino acids in position 2, 5, and 9, that peptide fragment can be bound on the surface of HLA-A3. So there are certain amino acids at certain positions that have to be conserved, because those are the ones which are actually going to be involved in binding to the peptide binding groove. But as far as the other ones are concerned, we don't care, basically. And these ones that have to be conserved are called the anchor residues, okay, for that HLA molecule. And they're called anchor residues because they anchor the peptide fragment to the peptide binding groove. So let me just discuss the structure of specifically these amino acids. Okay, so just to make it crystal clear. Right, so isoleucine we needed in position 2. Okay, so what's the structure of isoleucine? Okay, so isoleucine then, so you have an amino group here which will be bound to the amino acid upstream of it. Okay, then you have an alpha carbon here, okay, with a hydrogen off it, and then the structure of isoleucine is that you have a carbon, then a second carbon, and a third carbon like this, and then a methyl group sticking off here, okay, like so, and then hydrogens to saturate it. Okay, so this is the structure of isoleucine. So I stands for isoleucine. Okay, then we have the carboxylic acid group. Okay, and that will be bound to some other amino acids. Then you'll have the third amino acid, which I'll just put as the number 3 here, because we don't know what it is. I'll put 1 in as well over here. Okay, here's 1. Then you'll have number 4 here, and then you'll have 5. So here's the amino group with the hydrogen off it. Here's the alpha carbon. Now in position 5, what did we need? Well, we needed a phenylalanine. Okay, and the structure of phenylalanine is that you have a methylene group here, like so, with then a benzene ring off it here. Okay, so I'll just draw the benzene ring as a hexagon with a circle in it. Then you have a carboxylic acid group. Okay, so that's position 5. Then you have 6, 7 and 8, and 9 isn't going to fit over there, so we'll have to go on to the next row. Okay, so here's 8, and then in position 9, you then need to have an amino group, which is obviously the amino group of the amino acid. And then we have our alpha carbon with a hydrogen off, and then here we have lysine. Now the structure of lysine is that you have 4 methylene groups. Okay, so there's 1, here's 2, 3, and then the 4th one will then have an amino group coming off here. So this is the 4th methylene group, and then of this you then have the amino which is at the terminus of this 4-carbon R group. And then we have the intact carboxylic acid group, which will be involved in binding to this binding pocket, since we're talking about an HLA-A molecule, which is an example of an MHC class 1 molecule. Okay, so you have to have these 3 anchor residues which are involved in binding to portions of the peptide binding groove, and without them the peptide fragment won't attach to the peptide binding groove of the MHC molecule. Okay, and this goes for all of the MHC molecules, all 3 types of MHC class 2, all 3 types of MHC class 1, and all of the alleles of all of those 6 types will then all have different anchor residues that they need. Okay, so something about this HLA-DR4 is going to load peptide fragments. It will have a certain combination of anchor residues, and then it will load certain peptide fragments, which is going to then lead to rheumatoid arthritis, and we'll discuss that in our videos on the pathology of rheumatoid arthritis.